All right, so today's event, as you're all well aware, we're going to be talking about shark research around North America. Now, I'm going to start this off just with a really brief overview of some of the different options after high school. And that's because our guest speakers are going to be talking about different degrees that they've pursued. And I just want to make sure that everyone's roughly on the same page here. So after you finish high school, you have so many different options ahead of you. One of the options is to do the university track. So if you decide to go to university, the first thing you're going to be pursuing is something we call a bachelor's or an undergrad degree. So people have different names for it. And usually that's 4 years. You can do it in any topic under the sun pretty much. But if you think of the traditional STEM disciplines or fields, like I have my bachelor's degree in biology, you can do chemistry, physics, environmental science, I'm blanking, math and stats, languages. There's so many options, so many options. After that, another option is for you to do your master's. And usually for your master's, you're doing a research project. And usually this takes around 2 years. So I believe, Triana, you're in your master's right now, right?
Yeah.
Fantastic. After that, and though you don't have to keep doing more school is what I'm trying to say, but some people decide to. And for those people who decide to do even more school, they do a PhD. And Emily, you're a PhD student right now, aren't you? Yes, all right,
Correct.
So you have a PhD here, and that takes 5-ish years. And once again, that's a huge research project that you're devoting a ton of time to over your 5 years. And then after that, if you still can't get enough of school, you can do something called a postdoc. And I believe Brynn is doing her postdoc right now. Usually that takes around 2 years. So you might hear some of these terms today. I just wanted to lay this out so you're a little bit more familiar. All right, next up, what do we have? Okay, so we're talking about sharks today, but we're not just talking about sharks. So everybody's probably aware of what this animal is here. We've got a beautiful, gorgeous, great white shark. That's a shark. Over here, we have something that some of you might be familiar with. When I did my master's, I studied these absolutely gorgeous fish called skates. Emily also studies skates. We love them. Down here, we have a stingray, very closely related to our skates up here. And over to the side, we have an animal you may have never heard of. That is a sawfish. So the question here is, what connects all of these different animals? So that's the term for all of those animals. I know that's a weird looking word. I'll say it again, Elasmobranch. So you might hear that today. And one of the main features is that they all have a cartilaginous skeleton. So if you wiggle your nose around, if you wiggle your ears around, the reason you can bend them and not break them is because they're made of cartilage. So one of the things that unites all of these different fish species is that their skeletons are made entirely of cartilage. The only other thing that unites them, and of course I have shark experts here who can correct me if I'm wrong, is that they all have between 5 and 7 gill slits. So if you know what their gills are, they're these kind of slits that cover them. And between 5 and 7 is something else that unites them.
I am Jasmine, and I'm going to tell you a little bit about my journey into marine science. So there are lots of important organizations and institutions that helped me along my way. Most of them are here on this slide. So when I was really little, in elementary school and middle school, I did what's called in the United States magnet programs, which were geared towards science and math. One of them was called ALERT, and one of them was called TLC, the Learning Collaborative. So that's kind of where I first got my experiences, I guess, in science. And then I spent some time trying to figure out what I wanted to do, and I knew that I loved the ocean and fishing and all of that kind of stuff. And so my parents actually sent me to a camp called Marine Quest that is actually at UNC Wilmington, where Emily's at. And it is a super awesome camp. And it was at that camp that I figured out that you could do marine science as a career. And so I was kind of like, whoa, you mean I can get paid to play in the ocean with fish? This is awesome. And that was when I was, I think, 14 or so. So probably about the age of some of you guys. And I kind of was like, yes, okay, I want to do marine science. How do I do marine science? And so I decided that I wanted to go to college. And so I looked for a bunch of college programs that offered marine biology. And I ended up going to the College of Charleston, which is in South Carolina, which is on the east coast of the United States. And I had a blast there, and I really got to do a lot of different research topics. So I tried out a lot of different things. I did some microplastic stuff, which is like little itty bitty tiny plastic that's in our oceans, unfortunately. So I studied that a little bit. I did marine mammal stranding. So a stranding is whenever a marine mammal, for some reason, whether they're injured or sick or confused or whatever, they get up on the shore and they can't get themselves back in the ocean. And so I worked with the Marine Mammal Stranding Network that tries to get them back in the ocean and rescue them, or tries to rehabilitate them if they have some sort of issue. And then if they die, we try and figure out why they died. So I worked in what's called the necropsy lab, which is very similar to like autopsies on humans. So you do autopsies on humans, you do necropsies on animals. So that's basically where you just try and figure out, you dissect it and try and figure out why it died. And then, of course, I did my shark research. And so I kept coming back to the shark research. I would kind of venture off and say, oh, let me try this thing. And then I'd be like, nope, I like sharks better. And I'd come back. Oh, let me try this thing. Nope, I like sharks better. And then I would come back. So I did all kinds of stuff, tried everything that there was to try at my university, and I kept coming back to sharks. So I decided, yep, that's probably where I should be at. So I decided to do research on sharks for a career. I did a couple of what's called research experiences for undergraduates, which is funded through the National Science Foundation. I did one at College of Charleston at the Marine Lab, and I did one at the Smithsonian Environmental Research Center, which is in Maryland near DC in the United States. And I worked on some ocean acidification stuff there. And then after I did all of these research experiences and I kept coming back to sharks, I talked to my advisor and I was like, I want to do sharks. How do I do sharks? And he said, well, do you want to go to grad school? And I was like, yeah, I don't know what grad school is, but that sounds good. And so then he kind of explained to me what grad school was and the process. And so I decided to go and get my master's degree. I got my master's degree at Florida State University, and that's where I started working on sawfish. So my master's degree was fully funded by the National Science Foundation. Me and the National Science Foundation are tight. I do everything with the National Science Foundation, I realize. But they have a program called the Graduate Research Fellowship Program, or GRFP, and that funded my research and my tuition and paid me to go get my master's degree, which was super awesome. And I did that, started working on sawfish, got more into the ecology side, so studying how animals live and interact with the environment, versus what I was doing in my undergrad or my bachelor's degree, which was more focused on understanding the evolutionary history of sharks. So it was a lot of lab work, but when I transitioned to my master's degree, I decided I wanted to spend more time on boats fishing and out in the water. And so I transitioned from a kind of lab-based science to a field-based science. And then some other institutions that have been really influential, the American Elasmobranchs Society, which I joined 4 years ago. Yep, 4 years ago. And now currently I work at a place called Mote Marine Lab, and super awesome. It's actually a place that when I first got interested in sharks, I wanted to work at, because I found out about a phenomenal shark scientist whose name was Eugenie Clark. And she, like, Mote is her baby, and she like created Mote from basically nothing. And so I was like, oh, I want to go work at Mote, because Eugenie Clark was an awesome lady, and everything that she creates is, I'm sure, awesome. So I'm really excited that I get to work in a place that I have wanted to work at for a really long time. So here are some really fun photos that I kind of pulled from my research. So like I mentioned, when I was doing hammerhead sharks, I was studying how they're related to each other. So their evolutionary history, and I was building a phylogeny, which is basically like a family tree for sharks. Who's related to who? And that's what I was trying to figure out. And so I did that in a couple of different ways. The first way, I'm sure Emily's going to talk about some CT scanning as well, but I did CT scanning of sharks. So you can see over here, this is a picture of some sharks being CT scanned. And then I would take those scans, which would kind of be like a mess, kind of like a polar bear in a snowstorm. And I would have to do what's called digital segmentation, which is basically like doing a virtual dissection on the computer where you like manually go in and take out all of the important structures. So that's what I'm doing here on this computer. The other part of my research, so not only was I looking at the anatomy or how the sharks' bodies were built, but I was also looking at their DNA. And so I did DNA extractions and I analyzed their DNA. So lots of lab work, lots of really super-sized pipetting, and that was a fun time. So I got some pictures from here. The sharks that we CT scanned actually are museum specimens, so they have been preserved. So here's an example of one. And the really great thing about CT scanning a shark is you don't have to actually cut into it. So the museum specimens, I scan them, I send them back to the museum, and I haven't damaged them at all. So more people can keep using them. So that's why we use this method. Some more interesting photos I have here. So this is a video that kind of shows you how I do my scans. So there's like, it looks really complicated. Well, it is kind of complicated, but you have different pieces that are not the pieces that we're trying to get. So I'm going in and I'm trying to remove all of those pieces that I'm not trying to get right now. So you're gonna see me take out everything that's not the specific structure that I'm looking at. And so we're gonna zoom in on this little guy right here, and we're gonna get rid of everything else. And then you'll see me go in and do some really specific little dissections where I'm going to start to break apart all of the little pieces until I have just this structure remaining. So that's kind of how that process goes. Depending on how clear the scan is, this could take me as little as, I would say, 5 or 6 hours. If it's a really difficult scan, it might take like a week. So it kind of just depends on how fuzzy it is, how mushy it is. So like we mentioned, sharks are made of cartilage, and CT scans, these are the same CT scans that they use in hospitals. So they are specially developed to identify human bones. So sometimes they get a little confused when they're trying to differentiate between skin and cartilage because they're used to looking at bone. So sometimes they are a little more difficult to dissect than others. But that is the process in a nutshell. And then these are some examples of how these scans turn out when I'm all done. So we have I have an example on the left of what it looks like with the skin on. And then on the right is the same shark, the winghead shark, on the other side with the, where you can see all of the different pieces that I've segmented out here. And I have an example here in the middle. And I just have this example here because I want to point out that sometimes your shark had some lunch, and you get that in the CT scan. So sometimes you can actually see what was in their stomach when they died. So this guy has some lunch in there, which is pretty interesting. So I did all of that with the hammerhead sharks, but I also did a little bit of field work while I was at College of Charleston, and got some experience. And I also did some field work with my ocean acidification project while I was at the Smithsonian Environmental Research Center. So this is our little machine that we developed that would measure a lot of different things in the ocean's quality. So it was measuring pH, CO2 concentration, and all kinds of things. So sometimes in science, you get to be kind of an engineer and a plumber, and you got to build some things to, if they don't exist yet. And so this is just some of what I was doing. And I was doing some titrations to figure out the pH levels. I was looking at how many plankton were in the water. So I was doing some counts of that in a microscope. And then of course I have my sawfish research, which I do now. And so the sawfish, I mean, they're really small when they're born, and then they get to be like 16 feet long. And you're like, whoa, how did you get that big? Pretty crazy. These are critically endangered. This is the small-toothed sawfish. And I insert little tracking devices in them so that we can track where they're moving, so that we can find out where they're spending a lot of time, so we can make sure that we protect those areas. So yeah, that's what I do. I get to fish for these big old guys, which are super rare. So I'm really excited that I get to work with them. Most people go their whole lives, even if they do live in the area where the sawfish are, they go their whole lives without ever seeing one. So I feel very fortunate that I get to do my research on these guys. And then lastly, I recently formed an organization called Minorities in Shark Sciences. And this is a program for women of color interested in shark science. And I'm really excited about that. We just started it about a month ago. And so we are working on developing some programs. We're putting on a workshop for students in college or recently graduated that want some experience with sharks but haven't had the opportunity to get any. And so I'm really excited about that. And so if you guys ever want to look it up, we have a website, misselasmo.org. And that's me in a nutshell.
Okay, yeah. Excellent. So thank you, everyone, and thank you, Jasmine. It's a tough act to follow, so guys, Jasmine is awesome. So yeah, I'm gonna talk a bit today about how I went from a little kid studying minnows, this minnow is very unhappy, to doing my PhD where I study shark and skate brains. And so my educational path really began when I moved to the coast. So like Julia said, I'm from Atlanta, which is nowhere near the ocean. And I went to the University of North Carolina at Wilmington, where I did my undergraduate or bachelor's degree. And at the same time, and a little bit after my undergraduate degree, I also worked at the North Carolina Aquarium. And so I'll talk a little bit about that. I could talk all day about the aquarium, but I was really, really lucky there to do a lot of different projects and get my feet wet. So I did everything from rehab turtles, I trained alligators, I was able to dive in the big tanks. We went on a load of collection trips to get anything from frogs and snakes to sharks and stingrays. And I also was able to participate in a few different research projects. One down here was looking at southern stingray health. And we also looked at sand tiger shark reproduction, which was really fun. And I can answer any questions about working at an aquarium. But while I was at UNCW, I started my own research project looking at aging bonnethead shark pups using fancy word umbilicus morphology, which is essentially a complicated way to say that I studied shark belly buttons and how they changed after they were born. So those of you who don't know about bonnetheads, it's a small coastal species found anywhere from North Carolina down to Brazil. And in the wild, recently born sharks are aged based on their appearance of their belly button. But previously, this is more of a rough estimate because when you catch them in the wild, you don't know exactly when they're born. So what we did is we went out and collected wild pregnant bonnetheads, and we reared the pups from the day they were born into about 200 days after they were born. And so to do that, we took the sharks, flipped them over, shone a dive light on them, and then we just took a picture. And I focused again on the belly button, looking at the area of that scar and then how that changed throughout time. And so what we found was that the scars healed in pretty much the same way, and we could classify them into 5 different stages. So that first stage is where they still have a little bit of that umbilicus from mom left on their tummies. And then 2 is where it still has a more open area. 3 is where it pinches in the middle. It looks like a figure 8 scar. Then 4, it pinches down at the bottom, still has a little bit of open left at the top. And then 5 is just a really thin line at the bottom. And so what we did is we put an age range on those different stages to be able to estimate when they were born based on, again, how their belly button looks. And we're hoping to use this to, and the appearance of the belly button to find out where nursery habitats are, because if you catch a shark with one of these really early stages, you can estimate that they were probably born within the past 1 to 10 days, and they were probably born in the area that you collected them in. And another really fun part about this research was I was able to do a fancy microscopy technique, which is called SEM, or scanning electron microscopy, which is essentially just really, really high power microscopy. And so what we did is we cut this little area of the belly button, and then we were able to look at it. So eagle-eyed people out there can see that this is a stage 4. So there's still a little bit of open at the top. And so you can see at this bit near the tail that the scales are coming back in, but they're all really strangely shaped and they're not coming in the right direction. But then at the top near the head, there's still a little bit of open area where the scales have not come back in. And then if we zoom in to this bit up near the head, you can again see where there are no scales. So this is just scale-free epidermis, and that will eventually grow scales, but this is a time period when they haven't all come back. And then another really fun part of this, which was not related to wound healing at all, is when I was taking the picture one day, my friend and I just put a dive light underneath the shark, and we found that you could see each one of the individual ampullary canal pores, which at the base of those hold the ampullae of Lorenzini, which if some of you have heard of that before, they can allow sharks to detect weak electric fields. And so this was really fun for me because I was able to see that when you're doing research, sometimes the most interesting or the most beautiful parts are what you really didn't even plan on. And so this wasn't part of my research, but I love these photos and I think it's so cool to see those. So after I left UNCW, I graduated with my bachelor's in science and biology. I continued working at the aquarium looking at more wound healing in the Atlantic stingray. So we already know that sharks are super great at wound healing from previous research, just following the same shark after they had an injury. So you can see this is a black-tip reef shark that has a lovely little bite. And then after only about a month, you could barely even see the bite. But there is very little research prior to ours on sort of how the wound closes and how the tissue reforms. And especially what we were excited about was how the mucus covers the wound. So if you guys have ever touched a shark, which I think some of you have, you can feel they kind of are a little bit slimy after you're done touching them. They are rough because they have the scales, but they're also slimy. And that mucus helps protect the outside of their skin. And so we were interested in seeing how that mucus plays a part in their wound healing process. So what we did is from collaborators at Moat, thanks Jasmin and her crew, we looked at Atlantic stingrays and cut tiny, tiny little thin holes in the wings of the stingrays. And then we sectioned up the tissue really thin, stained it, and then took pictures with a microscope. And so this is what we found. So this is the piece of tissue that was not wounded. And then what we did was we cut a little square out of here. And then you can see in this pane, after 16 hours, this purple bit is actually the mucus. So you can see it just starts pouring into the wound. And then after 1 to 2 days, it's completely covered. And then over here at 35 days, you can see the top layer of skin is really thick. So it's growing over, it's healing, the tissue's growing back. So after only about 8 hours, what we saw is a huge spike in the mucus. And then after only a few hours, it covers the wound completely. And so this proves, that sharks are really great at healing. And a large part of that is due to their mucus just pouring over the wound, which allows them to have an infection-free healing, which I would love that, if I didn't have to worry about infections. So sharks and stingrays are very cool in their ability to heal. So after I finished that research, I was ready to go back to school, ready for more pain. So I started my PhD at UNCW and I joined the YOPAC Zombie Lab. So as the name suggests, we study brains. And part of my project is looking at the effects of increased temperature on brain development in 3 different Elasmobranchs species. And so I'm sure, as you all know, over the last century, sea surface temperatures have increased about 0.5 degree Celsius, but they're predicted to increase another 1 to 4 degrees by the end of the century. And changes in an animal's biological performance can change based on the environmental temperature, and it can impact a number of their life history traits. So it can impact their growth rate, and it can impact how many babies they have. It can also impact their behavior and their metabolism. So metabolism is really, really important for fishes, because in most cases, their metabolic rate is in large part determined by the temperature of the water. So if you have an increase in temperature, it's going to affect how they live and how they use their energy up. And another part of this story is that animals can alter their form and behavior with changes in their environment. So a lot of studies have shown that changing an animal's environment can also affect their brain. And if you have a reduction in your brain size, it's going to negatively impact your life. Right? So the reason that this is so important for fishes especially is because they grow forever. Oops, I moved this over. Okay, yeah, so they grow forever. And so they may be more affected by changes in their environment throughout their lives. Oops. All right, so not only do fishes grow forever, but their fastest brain growth occurs really early in life. And so this means that there might be a sensitive period during that time to changes in their environment. But we don't know if and or how climate change can affect this. So to study this, we looked at 3 different species, and they were reared in either normal temperature or high temperature. And so we looked at the little skate, which these were collected in the Gulf of Maine. Then we looked at the epaulet shark, so these were collected from Northern Australia. And then Port Jackson sharks, and these were collected from Southern Australia. And then we use MRI. So Jasmine talked about CT, and so I use CT, and I also use MRI. And this allows me to get really high resolution 3D images of these animals non-invasively. After I get these MRI scans, then I can use a special software to segment out the brain, just like Jasmine showed you, she segmented out the cartilaginous skeleton. And so this is, again, a scan of a little skate going through. So you can see the gills here. This is the brain, and you'll see the eyes in just a minute. You can see the spinal cord here. So this is a really amazing way that I can compare brains. And this is a scan of one of the epaulet sharks. So again, you can see the brain here, the eyes on the side, you can see the muscle. And I used, again, that brain 3D image to compare sharks that were reared in normal temperature versus sharks that were reared in warmer temperature. And what we found is actually that sharks that were reared in warmer temperatures have a smaller brain, and that brain regions that are more associated with learning, memory, and smell are all smaller in the sharks reared from warmer temperatures. Now, so taken together, these results mean that we really need to look at how different species found across different habitats and at different temperatures are all affected by increasing ocean temperatures, which may help us determine how fishes will fare in the changing oceans. So other projects I'm working on are looking at normal brain development in sharks, and then working on new methods to actually count the brain cells, the number of brain cells throughout development. And then after my PhD, I'll continue to study brain development and how environment can affect that. And then I also want to eventually become a professor, because one of my very favorite parts of science is having the opportunity to teach and work with the next generation of scientists. So I want to thank you guys for being here and inspiring me to continue doing my research. So this was done with a whole lot of help from a whole lot of different people. As Jasmine said, AES has been a huge part of that, the Yopac Zombie Lab, all of my advisors. So I've had the opportunity to have 2 wonderful female advisors. So that's been really wonderful. And so that is me.
A little kind of like tidbit also. Emily is really good at presenting. Just like in general, she's so good at presenting. As someone that's like watched her presentations at AES, she always phenomenal. So, you know, I love watching my friends present, you know.
And actually, Triana, just before you dive in, I will say, so maybe you girls have heard this term AES a few different times. And so when you're in university and you're doing research, sometimes there's like different groups or conferences that you go to. So AES stands for American Elasmobranchs Society. And basically it's a conference that happens once every year, and shark biologists from all over the world, there Triana has her name tag there, perfect. So shark biologists from all over the world meet together in this one spot to basically talk about the research, right? Everybody wants to know what everybody else is doing. And conferences are actually so much fun. So if you guys end up going into research and going to conferences, they're awesome. I met Emily at a conference like 5 or 6 years ago, and we are still friends. So there you go. Multi-purpose. You get to learn and you get to meet friends. Sorry, Triana, go right ahead. Thank you.
No, you're totally fine. I realized I probably should have expanded on AES instead of just saying it. But hello, everybody. Thank you so much for taking the time to be here. My name is Triana Arguedas Álvarez, and I am currently a master's student at El Colegio de la Frontera Sur, which is a Mexican research institute funded by the government. I am in one of 5 campuses in the south of Mexico. So we are in all the states that border Belize and Guatemala. So we're only found in the southern frontier states, which allows us to conduct research. We have people that work in Mayan communities that work with different animals in jungles, in mountains, in deserts, in the oceans, both Pacific and Atlantic. So we have a lot of really cool research at my school. And something that kind of doesn't happen very often here in the United States is that this is only for research. So we do not have undergraduate students. We are all postgraduates. We are all master's, doctoral, or postdoc students. And we all, almost like how Jasmin said, we get paid to conduct our research, which is really, really cool. And I didn't know that existed when I was younger. Especially, because just a little bit about me. I grew up in South Florida. I am a first-generation American, so I am Mexican. Both of my parents were born in Mexico and then moved to America. I love learning about all things nature and animals. So why do I bring up that I'm Mexican? It's actually something very important as part of my journey into science. I come from a very long line of teachers and engineers, and I'm actually the first biologist in my family, so that's really cool. I actually have a cousin who also is a marine biologist, but she actually works in Antarctica. So we're like very opposites, but we are like the only 2 biologists in our family. And it's really cool that we get to connect and kind of exchange really cool ideas. And another thing that I'd like to bring up is being first generation, I didn't have a whole lot of guidance with how the American university system worked or what I was really supposed to do, My parents, are very supportive and went on this journey with me and learn at the same time that I do. So I didn't know about all these conferences, all these mentorships, all these scholarships and programs like Terranaut Club, these amazing outreach programs. We didn't know anything about it, So I'm really thankful that my parents are learning right next to me, and now they can say that, they know all about AES and they love learning about sharks and, and especially in South Florida, because there's a bunch of sharks in South Florida. So they love, going out and saying, oh, there's a black tip, or, oh, there's a bonnet head or something. So it's really fun that it's kind of like a family affair, Okay, so my undergraduate work actually focused on nursery habitats. Again, my journey into science was kind of a stumble into science. I always knew I wanted to do research, but again, I didn't really have a whole lot of guidance. My dad wanted me to go into software programming or IT or, engineering, but that wasn't really my calling. And my mom really pushed me to keep pursuing research because she knew that's just like my personality type, is just always asking questions and trying to figure out like the answers to all these things, that are so amazing in the world. So I'm really thankful that I had an amazing, amazing mentor in undergrad, Jim Gilschleider, Dr. Jim Gilschleider. He works in toxicology, which is it's exactly what it sounds like. So toxins in the water that can harm the animals in the environment. He studies that. And he also studies growth and reproduction in sharks. But he also has a field survey. So every summer he takes 6 students out, 6 to 9, I think, students out every summer to go tag sharks as part of a long-standing survey program. And from that, I got to help continue a nursery research project that I actually got to present at a conference, which was really exciting as an undergrad, to be able to show everybody this really cool thing that you learned and get to tell everybody about, you know, this thing that really sparked your interest and, you know, something you're really passionate about. So here we have a cute little nurse shark that we caught. We also had the opportunity to go to the Bahamas and participate in some of their tagging efforts and research projects. So here we have an oceanic whitetip that is being, well, I was flipping it over to get it out of tonic immobility. So sometimes when you flip a shark over, they actually go into a kind of state of non-move, like they don't move, they're kind of frozen. And then you have to kind of flip them over and wiggle their tail. And then they wake up and then they swim away. So it's a really cool thing to experience and learn about. And then I'm really thankful for that. And in this last picture, in this last picture, we actually were also corralling stingrays. So we were literally chasing them around with nets, trying to get measurements and tagging and making sure that we kind of have an idea of what the population looks like in the Bahamas. And, you know, I'm really thankful that I had the opportunity to participate in these different projects because it kind of allowed me to better understand that how diverse shark science is. And similar to Jasmin, I had a bunch of different research opportunities. So I've worked with eels, oysters, dolphins, sea turtles. Oh, and then I'll show in the next slide, sawfish. So, you know, there's just something about sharks. They always call you back, you know, they're the coolest, weirdest fish, and you just, they're irresistible, you know, they're just so much fun. So something that I also got involved with is actually at a conference, I met a researcher who was also Mexican, is Mexican, and he was a very prominent fisheries scientist in Mexico, in the area that my school is in and where I'm conducting research. And thankfully I kept in contact with him and actually got the opportunity to work in an internship with him. So I helped set up, I got to move, well, I moved to Mexico for a couple months, and then I worked with him organizing permits because we were going into national protected marine areas. We were going into, we were doing research fishing. So it's like a whole different slew of permits and just maps and people you have to contact and government officials and stuff. So that definitely opened my eyes to how fisheries and just in general marine biology science works in different countries. And that kind of started to push me in the direction of fisheries. And for those of you who don't know, fisheries is basically a very, very, very multifaceted. So there's a lot of different things that go into fisheries. It's a very, very diverse field in marine biology, where we basically study how fish go from the ocean to your dinner plate. And they're all the stuff that goes in between. So all the biology, the ecology, the field work that you see, that you saw Emily and Jasmin talk about, all of that stuff, how does that affect how we regulate their fisheries, how much do the fish cost at the end of the day, what kind of gear is okay to work and try to capture these individuals for market sale, how they're sold, is it legal catch, is it illegal catch? So there's like so much that goes into fisheries. So learning about how different countries work in these economies, it really kind of sparked my interest, especially being from Florida and then in Mexico. I realized that we share the Gulf of Mexico. You know, we share the Gulf of Mexico with Cuba and Mexico. So how do you control the fish that get caught there? You know, who gets to say, oh, this is my fish and I'm going to keep it? Well, what about the fish that were born in a different country and then swim to Cuba or swim to Mexico or swim to the United States? So that's something that I'm really interested in. And working and living in Mexico actually really sparked my interest. And so here, and this was like this actually particular project was very out of my wheelhouse. But here, sawfish are supposed to be, or used to be endemic. So they used to only live, or not only live, but used to be very popular species in the Gulf of Mexico. So they used to be found everywhere. Like they were caught as bycatch, they were caught in sport. They were very, very abundant in the coasts of Mexico. But due to overfishing, it's suspected they're actually locally extinct. So with this researcher, I had the opportunity to practice an eDNA technique where you basically take samples of water, you filter them, and you do a bunch of really cool science that I'm not super well versed in, but basically it kind of gives you like a grocery list of all the animals that have passed through that water column. So it's really cool because it's kind of, it takes different DNA samples from the water and it allows us to know what animals are living in those waters, even though we're not fishing for them or we can't physically see them in that moment. So it was really cool to learn that research technique and kind of learn about how we are trying to work in conservation while using these really new innovative ideas. And then here I have a picture of just some sawfish data that I had to work with. And I also put it because as much fun as field work is, at the end of the day, a lot of scientists, we do have a lot of time. We do spend a lot of time on the computer writing and trying to figure out if our graphs look right, if our pictures are the best quality, if our data makes sense. So, you know, it is glamorous, but then there's also a little bit of computer work that we have to do. So this was the gap year. So I decided after undergrad to take a break and try to do something at my own pace. So it was when I moved to Mexico and then, you know, trying to figure out what I wanted to do, where I wanted to go to school. And because of this trip, I actually got to meet my now advisor. He, again, is based in the Campeche campus in the south of Mexico, located in the Yucatan Peninsula. And we work in shark fisheries. So again, going back to fisheries, in the United States, we have sustainable fisheries by species. In a lot of developing nations, that species-specific identification does not exist. So for example, in Mexico, there are two different size classes. There's gazon, which are sharks that are caught regardless of species between 0 to 150 centimeters. And then anything above 150 centimeters are classified as tiburon, which are large sharks. Doesn't matter the species. So this complicates things when we're trying to figure out if we are overfishing, if there are species that are maybe endangered in local waters, or if there's migratory patterns that we are not aware of, or, you know, all just these different questions and these different holes in information that we really should figure out if we want to take care of our oceans. So my current research focuses on the bull shark, which is a very charismatic, a very large, easily identifiable large shark that lives, it's very abundant. You can find it almost anywhere. It's a species, thankfully, that's classified as nearly threatened, so they're not as bad off as sawfish, but they're also not, you know, they could do some good close eye monitoring. So here, I am sorry, but here's a small species of shark. This is a black nose shark that is commercially sold in the fish markets in Mexico. And a really cool thing that I learned is in the Yucatan Peninsula and actually most of Mexico, sharks are consumed in dishes. So they have a bunch of different dishes that are culturally and historically important to Mexican culture. And it was really cool to learn about the significance that sharks have, not only in a biological sense, but also an economic and social sense, because again, fishing is a very large portion of employment. It provides food security, especially for a developing nation such as Mexico. And then here we have a very standard fishing vessel. Most of them are gill nets, monofilament, depending on the target species, even though they're technically multi-specific. So they go out, they set their nets, and then whatever gets caught in the net is what they sell at the market. So it's not, again, it's not by species, it's by size. And it's hard because bull sharks, for example, when they're babies, they're under 150 centimeters. So they can be classified in both size classes. So that complicates things a little bit. But thankfully, we've been able to figure out with my advisor ways that we can try and figure out exactly how many bull sharks were being caught across 50 years. So we have government data that we use. I've been doing literature research. So I go in and I look for scientific articles and feces. So previous projects done by scientists in their undergrad or graduate programs that have very specific details about bull shark catch at a very specific point in time in a very specific part of Mexico, which allows us to kind of take a snapshot of what fishing looked like in that time period. So being able to take that information and apply it across 50 years is really, really helpful, because now we can kind of use this technique to figure out if our bull shark population is stable, if maybe they're migrating, using information again from Texas, Alabama, also information from Cuba, because you know, we have states that border the United States, like Mexican states that border the American states, and then the peninsula is super close to Cuba. So, you know, using that kind of mesh of data, it's gonna let us kind of peek into how the population is doing, if we need more species-specific studies for bull sharks or other economically important species in Mexico, and kind of take into account what the different fishing economy and practices look like in different countries. Because again, going back to an original, my original statement, we can't say, oh, well, this shark was born in Louisiana, so it's American now. That's, you know, that's not how it works. Sharks don't like get a little passport and say, well, I was born in America, so I'm going to stay here. But they like
That'd
to
be really cute if they did. Sorry, just a side
right?
note.
like to travel. So how do we figure out where these sharks are going? Who do they belong to? What are they doing in these waters? Are they reproducing in these waters? Are they being affected by climate change? Are fishing practices changing? Are government regulations changing? So again, fisheries is like a catch-all of like a million different things. But I really enjoy my research because it kind of, I feel like a detective going back in time and piecing together all these really cool pieces of data that are kind of hidden across all states. So I feel like, you know, like just on the computer looking for theses and trying to figure out, 3 bull sharks were caught in Tamaulipas in 1985. I wonder how that's going to look like when I apply this, and do that. So I enjoy my research. I think it's really, really cool. And again, I love learning about how different countries manage their fisheries and how maybe at some point, sorry, my dog just came in. I really like learning about how different countries manage their oceans, you know, because not, you know, one way isn't the right way for all countries. So I think it's a good opportunity to learn and hopefully go into cooperative management. So, you know, we can all share the oceans and take care of the oceans, you know? So with that, again, at the end, if we have any questions, please feel free, you know, at some other point, if I didn't get to your question or you come up with a question later, I have my email address and Julie can totally pass it along.
Yep.
And with that, I yield my time.
Thank you so much, Triana. And I'm so happy actually to have you talking about fisheries and talking about how we manage different fish, because it's a different perspective from our other guest speakers for sure. And that's something that we don't always think about, you know. Of course, we don't eat a lot of sharks in Canada. I don't think probably any of our guests have eaten a shark before, but it is worth wondering when a piece of fish ends up on our dinner plate, how did it get there? And that's something that I think leads to a whole different set of millions and millions of questions. So yeah, fisheries science is really, really fascinating. And because there's, you know, thousands upon thousands of species of fish in the ocean, you could be a fisheries scientist and work with, I mean, any number of them. But it's so fascinating that, of course, you studied sharks. So thank you so much for sharing.
No, thank you.
Yeah, thank you all for being here today. My name is Brynn Devine. I'm a postdoctoral research fellow. So I'm based here in Windsor, Ontario at the University of Windsor. But I'm actually funded and a participant of this program called Liber Euro. So they're a big national organization that funds research that addresses conservation concerns in Canada. So I was fortunate enough to be selected in 2019 to be a part of that fellowship team. So they're the ones supporting my shark research. So why do research? Just a few of the many things I love about research. I love learning about fish. Fish are my jam. I really love fish. I love meeting new fish and getting to go in the ocean or go on a boat and interact with fish that I've maybe only ever seen in a book or on TV. I love working in the field, if it's scuba diving or being on a boat, maybe it's camping and just working at a small boat. This last one sounds a little cheesy, maybe a little cliche, but exploring the unknown. I think my favorite part of research is just, I mean, there's so many things we don't know about the ocean, but finding a question, finding the best method, experiment to do to address that question, and hopefully discover something new, contribute something new to science. So those are kind of the things I love about research and being a researcher. So I'm pretty new to shark science. Before I studied sharks, I studied a lot of other fish. So I'm originally from Texas. I did my undergrad at Texas A&M University. And there I started my fish research. So I looked at blue marlin larvae. So blue marlin are these giant sport fish, like 5 meters long, these giant open ocean predators. But I actually looked at their little larvae. They're only about 5 millimeters long. You can set them on the tip of your finger. They're very tiny, but I looked at age and growth to try to figure out where nursery habitats are in the Gulf of Mexico. For there, I moved on to coral reef fish. I actually moved to Australia for my master's degree. So I did my master's at James Cook University in Queensland, Australia. So I got to work on the Great Barrier Reef, which was really, really cool. I looked at a lot of different reef fish to try to see how climate change and specifically ocean acidification, how it could impact fish behavior. From there, I made my way to Canada, where I am now, and I moved into the deep sea realm. So I worked for my PhD that I just finished last year at Memorial University in Newfoundland. I got to work with a lot of really cool, kind of creepy deep sea fish, looking at their distributions in the North Atlantic Ocean. And it's here that I was first introduced to sharks and the shark species that has now become my favorite and that I'm working on today, which is the Greenland shark. It's a really cool deep sea cold water polar shark. So I've been working on them for about 5 years now. So I'm just going to go through quickly what I've done with this species in the past, and then I'll touch a bit on what I'm doing with this shark today. So for those of you who aren't familiar with Greenland shark, I know they're not the one you see in Shark Week episodes, so they're not super known to everyone. So if you're not, they are a member of the family Salmonidosidae, so they're known as sleeper sharks. As the name might suggest, they're known for their extremely sluggish behavior. They're actually the slowest swimming fish in the ocean, literally, so very, very sleepy, very slow. They're found throughout the cold waters of the North Atlantic and Arctic Oceans. And I'm just going to hit you with a few fun Greenland shark facts because there are some really interesting things going on with this species. So they are a deep water shark. They've been observed at depths down to 2,200 meters, but they've also been seen swimming at the surface. So for a deep water shark, that's kind of unusual that they really use the whole water column. It's pretty cool. Most people don't think of them as being as big as great whites, but they actually do grow 6 meters or more. So they're one of the larger sharks in the world and certainly one of the largest sharks in Canada. I guess big news item with them is that we now think that they are the oldest living vertebrate on the planet. We think that they could live up to 400 years or more. They likely don't even start reproducing until they're 150 years old, which is pretty crazy. And then this last little fact, it might be kind of gross for some. I think it's kind of cool, but nearly all Greenland sharks are infected with this eyeball parasite. This little copepod that attaches to their retina, but it makes them functionally blind. So most Greenland sharks have extremely poor vision, maybe only some light sensitivity, but they live really deep down. So there's not a lot of light anyways, and they have a really great sense of smell like a lot of sharks. So they seem to be doing okay, even though they can't really see anything because of their eyeball parasites. So the research that I did on this species in the past was very hands-off. Unlike all of these other amazing shark researchers that really got in there and got to handle these sharks, I did most of my work through a camera lens. So I used this sampling technique called baited remote underwater video, or BRUV surveys. They are becoming increasingly popular. You have programs like Global Finprint that put these cameras down across the world to try to look at shark abundance and measure their populations. But it hasn't really been done that much in the Arctic. So for part of my research for my PhD, I had this big camera frame. Basically, it's just a camera, a light, a battery, and we pop some bait. We use squid in our camera deployments, put it on a bait arm, and then we put it in 5 sites throughout Nunavut in the Canadian Arctic archipelago to try to see what's there and specifically how many sharks might be there. So I'm going to try to share some videos here so you can see kind of what we saw. Hopefully it works. Yeah, so we learned some really interesting things from these videos. So in some of the places we would put the camera down and within minutes it would be this. We would have several sharks swimming around. It seemed like there were lots of sharks in this area. Other places we put the camera down and we could watch for hours and hours. Maybe one shark would show up, maybe none at all. So we really saw that there are big differences between these different areas that we sampled in terms of where sharks might be aggregating more, habitats that might be more important. Most of the sharks have really unique markings. So much like they do in identifying whales by their tail or great white sharks, these scar patterns made it really easy to distinguish individuals. So I was able to get a good count on the number of sharks that were in these different areas. So it gave us some insight into the local abundance in different sites. We also know what depth and temperature these cameras were at, habitats they are using, maybe what temperatures or depths they prefer. And then we also saw one more cool thing. So we don't really know much about smaller sharks, but in one particular site in this place called Scott Inlet, almost all of the sharks we saw were about this size. So this little guy is only like a meter long. And again, nearly all of the sharks at that one spot were very small juveniles. So we learned about where sharks are, where they aren't, but also where the smaller babies are. I know, so cute, they're so cute. Yeah, so we learned a lot just from those camera deployments. So it's really fun to see them swimming around in their natural habitat. So I'm still working with Greenland sharks, but I've taken a very different focus now in the research I'm doing. So I'm looking at bycatch. I know others mentioned fisheries. Bycatch is obviously a huge issue globally. Millions of sharks are caught as bycatch, and Greenland shark aren't any different. There's a lot of fisheries that we have in the Eastern Canadian Arctic, and Greenland sharks are very susceptible. So we have long-line gill nets and trawl fisheries, and Greenland sharks are caught in all of these fisheries. There's not a market for them, so all of the sharks are returned. Yeah.
Sorry, do you just want to explain really quickly what bycatch is? Because I think we might have some people that don't know. Perfect, thank you.
Sure, yeah. So bycatch really just means any non-targeted fish. So say you're fishing for turbot or shrimp. You may catch a lot of turbot and shrimp, but bycatch would be all the other things that you get in your net that maybe you weren't trying to catch. So again, these sharks are really slow moving. They probably, it's very hard for them to get out of the way of a moving trawl. So they often get swept up in these fisheries. So they're not trying to catch the sharks, but they do catch quite a few of these sharks. So again, they're all discarded back to the ocean, but as you can see from these photos, which are a bit hard to look at, but they do get some injuries, and it's probably quite stressful being pulled up from these depths and hauled onto a boat. So we really don't know how many sharks survive when they are returned back to the water. So for the research I'm doing right now, we're really trying to address this question and figure out what the fisheries mortality rate is, or how many sharks might die because of these interactions with fishing gear. So to do this, we're going to use these things called pop-off satellite tags. So it's this little black tag. We'll attach it to the dorsal fin of the shark whenever they're caught in bycatch. Then whenever they're returned back to the ocean, we can monitor their behavior for, say, 3 months after their return to the water, and we'll be able to determine whether or not they survived that encounter with fishing gear. So these tags are really cool. They collect a lot of really interesting, valuable information on movement and behavior. And then whenever you can program them. So say in 3 months, we'll say pop off the shark. The tag will pop off the shark. It'll float to the surface, and then this long, skinny antenna will relay all of the data to satellites, and then we can download the data and analyze it. So it's a really cool piece of technology. You can just stick it on a shark and it can tell you some really cool things. So from that, we'll be able to get an idea of how many sharks survive, or how many might die when they're encountered as bycatch. We can also get some other really cool information. So they do record depth and temperature throughout the entire time. So this is just an example of a tag that was on a Greenland shark for 4 months. So you can see it does some really deep dives. It changes maybe the temperature that it's occupying. So you can get some cool data on what types of habitats they're using. And hopefully that can help direct our fisheries to maybe catch less whenever we're fishing up north. So next steps in my career, I really hope to keep working with Greenland shark. I'm hoping we can find ways to reduce bycatch. And there may be really simple changes in how we fish. Maybe it's modifying a trawl to have a grate or something that keeps the sharks from getting in the net, or maybe it's changing the timing or the area that we fish that could really reduce the number of Greenland sharks that are caught as bycatch. There might be ways that we can improve how we handle or release sharks that can increase their chance of survival. And then I'd really like to improve reporting. So just making sure that when we do catch a Greenland shark as bycatch, we collect as much data as we can from that individual. I mentioned before, I love exploring unknowns with reefs in terms of how many things we still have left to explore. There's so many unknowns that remain with Greenland shark, and it's surprising given how large they are. We really know very little about them. So we have no idea how many there are out there. We don't know where they mate or where they pup, how many pups they have. We don't know how often they feed or their movement. And we don't really know their role in Arctic ecosystems. I mean, we think of them as a scavenging species, but there's also evidence they may be more of an apex predator like a lot of other shark species. So tons of questions that still need to be addressed. So I hope moving forward in my career, I can continue to be a very active researcher in the field and try to solve some of these mysteries related to this species. With that, I just want to thank you again, everyone, for participating. I know it was the last talk. Thanks for hanging on to the end. I'm moderately active on Twitter, and I at least try to keep it updated on what's happening with my research. So if, I don't know if any of you are on Twitter but want to check in, I'll try to keep everyone updated on what's happening in the Greenland shark world. Thank you.
Thank you so much, Brynn. So I think something that really stood out to me from your work here is I think often, or definitely when I was a kid and even when I was an undergrad, I thought that science had a lot more figured out than we actually do. I don't know if you other researchers agree with me there, but you kind of go like, there's so many smart people in the world. Like obviously there's like dozens of people studying great white sharks and we must know everything about them. But it's really the opposite. I think once you get into a research field, you realize that there are these huge gaping holes of information we just do not know. And Greenland sharks is a perfect example of that. And what you were saying, Brynn, about how we think that they can grow to be hundreds of years old, we only discovered that in what, the last 2 years or so?
Yeah, just the past few years. That's relatively new information, which is crazy. I mean, they're huge and they're pretty common in the Arctic. You think we would know a lot more about them.
When you were the age of our participants here, was there a specific subject in school that you weren't really good at? Were you always good at science is kind of what I'm trying to get to.
So I was, and kind of still am, really bad at math. Like, it's a problem. I have issues doing very basic things. So yeah, my hardest subject in school was geometry. Oh man, I spent all of middle school in my teacher's class after school because I couldn't, like, she would teach a whole class and I would be like, I don't know what you just said. None of that made sense to me. So yeah, not good at math. So now I'm really good at coding because I'm so bad at doing math that I would rather code and teach a computer how to do it so it does it for me. So that's like how I level it out. But if I had to do math on the fly, like I've taught classes before as a TA, and I do all of my equations ahead of time because doing math on the fly is never going to work for me because I'm so bad at it. I cannot hold numbers in my head. I don't know what's happening most of the time. Yeah, math is my weakest subject, which I think surprises people because they're like, you're a scientist. And I'm like, there are ways around it. I just train my computers to do the math for me.
Well, that's exactly what I'm trying to get to here is that to be a scientist, you don't have to be exceptional at every single science subject. You can always work your way around it. I had friends who really wanted to do a biology degree, but they said, I suck at chemistry. I hate chemistry. I don't want to do these 2 chemistry classes that I would have to do to get my biology degree. And sometimes you really just have to suffer through something to kind of see the bigger picture. So yeah, thank you for sharing that, Jasmin. I think there's probably a lot of us who wish we were better at math than we actually are. Triana, yeah.
So I'm not trying to like brag because I don't.
Oh, go ahead. You can brag.
But so again, my dad is an engineer. Like this man can do calculus in his head. He can do geometry in his head. He can find like the angles of everything like in his mind. Like he doesn't even need a piece of paper or pencil. And so in college, when I started taking physics and chemistry and all this kind of stuff, I started to feel a lot of pressure saying like, well, why am I not good at this? Why can't I do this? Like, you know, and so I really struggled with, for example, I took physics 4 times and I took Chem 2 twice. And I almost failed calculus, but I was in my professor's office like every single day for hours and hours and hours just doing practice problems. And I mean, thankfully we have, you know, I was very grateful to have professors that really took the time and cared about my success. So, you know, I definitely encourage you guys to not get overwhelmed or get down on yourselves because you are smart enough. It's you learn, you just learn how you learn, because everyone learns in a different way. And as we get older, we start to figure out, oh, well, I know that flashcards are really good for me. Or, oh, I know that I have to draw it. I know I have to write it 100 times. So as we get older, we start to figure out how we are successful. So, you know, maybe it took me 4 times to figure out how to be successful in physics, but looking back now, I'm really thankful that I stumbled a little bit, but now I feel, you know, that I was able to overcome those little obstacles, and I'm proud of persevering and, you know, working around it like Jasmin does, you know, like there's ways to work around it. There's ways, so she knows that she has to do the math ahead of time. Like you know, I know that if I stare at the computer for too long and I can't figure out why my formulas aren't working, I just need to take a step back and then come back and you know, fresh eyes, you know, like we all figure it out. And that's the whole process of being a scientist. You also learn how to be a scientist. You don't just wake up one day and you're just good at science, you know?
Yeah, everyone has to work at it. Absolutely. That's a really important point. Thank you for sharing that. And Brynn or Emily, did you have anything you wanted to add there?
Yeah, math is terrible.
Yeah, right. So same with Trey. I had really hard time with physics, and now I use it all the time for MRI. MRI is all based on physics, so it's something I've definitely had to learn as an adult. And another thing that I've also had to learn, we did not have coding growing up. This is a new phenomenon with everyone's age now, but we didn't have coding classes. We didn't have, I mean, we had typing classes, but that was the extent of our computer skills. So, you know, I've definitely had to learn about coding as an adult. And so if you guys have the opportunity to practice coding or play around with it or take a class in school, I would say take it, because it is the way of the future. It is the science. And so coding is going to be really important for you guys.